Skeletal muscles are a critically important part of our physiology. Of course, when they contract, they move our skeleton to help us walk, talk, run, jump, and more. Yet they also help us stabilize our joints and maintain our body posture. Skeletal muscles also generate heat and are storage sites for many nutrients. In this video, I will talk about how skeletal muscle is structured, starting at a macroscopic or whole muscle level, and finishing up at a microscopic or cellular level. I will not talk about how muscles are activated or how they contract, as I've done so in another video. So please see that video if you want to learn about skeletal muscle physiology. First, what we can see here is a whole muscle. Muscles, although this is not always the case, attach to our skeleton via tendons. We see whole muscles when looking at anatomy diagrams in textbooks and cadavers for those of you that have access to anatomy labs. Of course, there are many different muscles that come in different sizes and shapes all throughout our body. Yet, whole muscles are made up of many cells, which are called muscle cells or fibers, or myocytes. And these are arranged in a particular way. Typically, muscle cells are very long, but are bundled up together into something we call muscle fascicles. The orientation of muscle fascicles helps determine the force generating capacity of muscle. So, what we can see here is a whole skeletal muscle that is made up of many muscle fascicles, which are made up of many muscle fibers or cells. Let's now think about the components of, or what makes up, individual skeletal muscle cells. We know that cells contain organelles. An organelle within skeletal muscle cells that is actually so critical for muscle contraction that its absence will mean that muscles will not contract at all is the myofibril. Skeletal muscle cells are comprised of many myofibrils arranged in parallel to one another. Myofibrils are made up of three key contractile proteins, which we call thick, thin, and elastic filaments. These contractile proteins are called myofilaments, and the word myofilament shouldn't be confused or used interchangeably with myofibrils. Myofibrils are made up of many myofilament proteins. So, we've now established the hierarchy of muscle structure. Whole skeletal muscles are made up of fascicles, which comprise many muscle cells or fibers, which are made up of myofibrils. And myofibrils are made up of contractile proteins called myofilaments. Let's think about these myofilaments in more detail. If we were to zoom in on a single myofibril, which will span the entire length of the very long muscle fibers, we can see that there are repeating segments within the myofibril. These repeating segments of the myofibril are called sarcomeres, which are arranged in series. Sarcomeres are the functional unit for muscle contraction, and myofilaments are arranged in a particular way to give rise to sarcomeres, and it is the myofilaments within sarcomeres that cause muscle contraction. Let's zoom into one sarcomere to better visualize its individual components and to better understand myofilaments. Now, as I mentioned before, myofilaments can either be thick, thin, or elastic. Thick filaments are primarily composed of myosin proteins. Thin filaments are primarily comprised of actin proteins, and the elastic components are made up of a protein called titin. Now I've drawn myosin, actin, and titin here, and how they look in the sarcomere. Myosin is in yellow, actin is in blue, and titin is drawn in red. Sarcomeres and their myofilaments are what gives skeletal muscle its striated appearance. We can see that the myosin and actin overlap here a tiny bit. This overlap is important as myosin and actin interact during muscle contraction to shorten the sarcomere. So this little bit of overlap can turn into a lot of overlap as myosin pulls on or slides along actin to shorten the sarcomere, and eventually the whole muscle if we have many sarcomeres shortening. Titin essentially helps hold myosin in place. When looking at a sarcomere, you may also notice there are many parts of it that are labeled as lines, bands, zones, or discs. Let's quickly break this down. A single sarcomere is from Z disc to Z disc, or sometimes called Z lines. The middle of a single sarcomere is the M line. Myosin pulls actin towards the M line during muscle contraction, which shortens the sarcomere. The A band is the length of a single thick filament. Note, A bands contain both thick and thin filament overlaps. The I band is the region of the sarcomere where there is no thick filament, only thin filament. And lastly, the H shown is the part of the sarcomere where there is only thick filament, but no thin filament. I hope you now know a bit more about how skeletal muscle is structured, and thanks very much for watching.